because that's not correct. We're going to look at Hardy-Weinberg today. All right. Hardy-Weinberg is a theory and an equation. If you're a math person, give me a smiley face in the poll. How do you feel about math? I'll have to celebrate. How do you feel about math? I hate math. I love science, but I do not have the math and science mind. Yeah, I don't like math. So I tried to make this equation, in theory, as least mathematical as possible for you today. All right. But let's start with our goals. Could someone read our goals for us today? What are we going to do in biology this Valentine's Day? Who's in the house that will turn on their microphone and read to us our goals?
Good morning, gentlemen.
Hey there, JB.
What exactly is an allele frequency within any population? And in the equation P plus Q equals 1, what do the letters mean?
Awesome. All right, so let's start with this. What is an allele? Let's do a Monday morning wake up. Happy Valentine's Day, Peyton. What is an allele? Part of DNA, part of a gene. Good. Allele codes for one trait, all right? And it's a part of the code for one trait. All right? What do we use to represent an allele? Thing in a gene that does the thing. Yes. We use something. We don't use numbers to represent alleles. We have two types of alleles. What are the two types of alleles you can have? Maybe that will help. Types of alleles. Good. They're represented by letters. There's a dominant and a recessive. Ooh, good. X and a Y. That would be those are our gender determining alleles. You're absolutely right. But we can use any letter. We can use a capital A to represent a dominant trait. You could use a capital T, capital P, capital C. I'm just pulling random letters, right? Any letter of the alphabet with a capital letter is going to represent our dominant traits of a gene or of a organism. What represents the lowercase? Give me an example in chat. A recessive allele. Give me an example. Oh, blue eyes. Good. Well, just give me any lowercase letter in chat. Flood me. Good. Lowercase b, lowercase t, lowercase y. Anything lowercase. What if I gave you this genotype? All right. We're going to play that game. What's that game? I say, nope, can't even come up with it. It's like not even on the tip of my tongue. It's so far lost in my mind. Big A, big A. If I gave you that genotype, how could you label that or describe it using biology words? Good, dominant, but there are two of them. What if something is two? Yes, Kaden. Homozygous dominant, right? Homozygous meaning the alleles are the same. Homozygous, the alleles are the same. Kaden, that makes me so excited. Thank you. What if I put on the board this? What would you Big A, little a. How would you describe that? It's all right, no judgment on the spelling. Good, heterozygous. You don't even have to say dominant, but I mean, truly, the dominant trait would be the one that's expressed. Perfect. Hetero meaning different. So you've got homozygous dominant, you've got heterozygous. Now put in chat, we're going to flip it. I want you to put in chat what the allele combination would be for someone who is homozygous recessive. What would your genotype look like? Your genotype look like if you are homozygous? Yes, those are rock stars. Little a, or little letter. Homozygous meaning the same, recessive meaning small letter. Awesome. 100,000% that in April when we take these end of course tests for biology, that will be on there. Homozygous dominant, homozygous recessive, heterozygous, 1,000% that will be there. All right. Here's also another little plug. Mrs. Sheets, she used to work with me last year when I was teaching in credit recovery, but she is awesome and she is doing help sessions once a week now for any biology student that would like to start preparing for that test. All right, it's going to cover everything we've learned in biology this year, but it's going to be the main points, okay? But she's going to start reviewing for that test with you. So if you're interested in attending that or you see that on your Class Connect schedule, know that it comes highly recommended by Mrs. Rittenhouse. Awesome. So we know what an allele is. What is a population? This is more recent. What is a population? Population is what? Can a population be pigs, cows, and chickens? You're right. It's a group of living organisms, good, right, of the same species that reside together in a certain geographic location, right? So I can't say all chickens everywhere are a part of the same population, because that's not true. They're a part of the same species, but they're not a part of the same population. A population, yes, good, Amira. That they're gonna all be in a certain location. So I could have a chicken population where I'm at in Piqua, Ohio. All right. But Autumn, where are you from? Where do you live, Autumn? Where do you live, Izzy? Like what city? Kent. Alex lives in Kent. So if Alex has a bunch of chicken in Kent and Izzy has a bunch of chickens in Cincy, all right, they aren't going to be part of the same population. They're part of the same species. So we're going to look at a group of organisms today in the same area, all right, the same city, and we're going to look at the frequency of their a genetic trait, all right, one specific characteristic. So what could be a specific characteristic of a chicken that we could be looking at in a population? Like you could look at a chicken, like the color, or whether it's spotted. I don't know a lot else about chickens, okay? We'll just say the color. How long their legs are, that could be true. Good. Feathers. So that's what we're doing today, all right? So, we're going to look at these purple crested guys for a minute. Where is our dominant trait? What is our dominant trait in this exercise? Somebody put the dominant trait up here. Type it in for me. Good. Madison, do you mind writing that up there for me? And the recessive trait. So we've got the dominant trait of the purple crest. Oh, you don't have tools? I'm sorry. I don't know how my default permissions got a whack. There you go. You should have them now. I know we killed them off last week and they're back. All right, so we've got our purple crest is represented by a lowercase. Oh, capital F. Green crest is represented by what? Our lowercase. All right, so the very first thing that we do in this population identification is you have to determine, like if we were looking at chicken color or leg length, which gene is responsible for it in our DNA. So we've got to look in the DNA, which we can do with gel electrophoresis, right? Gel electrophoresis is basically putting your DNA on an electrode and being able to pull it out segment by segment based on length. All right. Something they can also do that is part of like crime scene investigation. So we can actually do this with biotechnology, is looking at our DNA. So we're going to look at a trait and we can actually pull that apart for studying purposes. All right. So for every set of alleles or traits, there's going to be a frequency. And a frequency just means what? Frequency is a fancy word for what? How much? Yeah, how long? How often something happens. All right. So like my daughter collected those little what are they called? Not not Hatchimals. Well, they were Hatchimals, but they're Shopkins. You know those little plastic things? They come in the blind bags, and you like undo your blind bag, and you pull them out, and they are all like little whatever. But she would look at them, and she'd be like, "Mom, this one's super rare." Like she would look at the paper, and she'd be like so excited because whatever, it was kind of like the Bengals going to the Super Bowl. Like, why did we want them to win? Well, because it didn't happen very often that we even get that far. So, what is the frequency? Well, you could say, well, like any team Tom Brady's on usually gets to the Super Bowl at some point, or these other common occurring teams that get to the big games or the playoff games. Hey, we were 3 points. We were winning the Super Bowl until a minute 35. We were left in the game. The Bengals were winning until the last minute and 35 seconds. So, can't make fun of them. All right, but I'm saying all this to put commonalities into your mind. Think of something in your mind that is rare. All right, that would have a very not like have it would not have a high frequency in a population. It would be like if you spotted a pigeon with this wing type, you just saw a rare occasion. All right, I'm not a bird watcher, but I imagine if you saw a rare, like, no, the Bengals didn't win. They lost in the last minute of the game. So anyway, if you saw a bird that was rare and you loved watching birds, I think you would be super pumped if you saw these super rare markings. All right. Versus guys like maybe this that is like they're flying around all over the place and have a not very difficult to see variation in their genes. All right. So we're going to keep moving. 4 times 6 since I promised to let you out early. Let's look at this, okay? Hardy-Weinberg is an equation that we use to determine the allele frequency or the how often you see a characteristic of an organism in a population. All right. So if we're looking at a population of coyotes or chickens or whatever, we're going to calculate how often you see that allele in that population nearest you. All right. It's like we talked about, a population is a small group of animals that are the same that lives in a similar geographic area. So the P, all right, we know there's only 2 alleles for any type of characteristic, right? We get one from mom, one from dad. It's either a dominant or a recessive gene. All right, so P is going to be the frequency of a dominant allele. So everyone say P equals dominant in chat, all right? Because the chat might be getting away from us. P equals dominant. See, I told you I tried to make it as least mathematical as possible. Right, bringing these little fluffy things, and you actually don't want to know how long I spent trying to find a good graphic for this, all right? P equals dominant. When we're looking at calculating allele frequency, we know there's only 2 types. So we're going to have 2 different factors that we're looking at. P is the dominant allele, Q is the recessive allele. So everybody put Q equals recessive. So if you are taking the end of the year biology test and it asks you what the P stands for and the Hardy-Weinberg equation, I want you to at least think this. Well, I know that there's only 2 types of alleles, so I don't want you to guess anything other than dominant or recessive. Okay? It's all right if you can't spell, no judgment. All right? And then I want you to think back to these little fuzzy guys. P equals dominant, Q equals recessive. And maybe we can say that the little fancy part of the end of the Q looks like a lowercase r. I don't know if that helps. All right. Or P comes first in the equation, just like a dominant letter, or you have a capital letter at the beginning of your sentence. Also, look at these. The P is big in the equation, right? We know lower uppercase letters stand for a dominant trait, right? So we can say, okay, the P is dominant. It represents the dominant allele. Q equals recessive. That's one of our main takeaways from the lesson today, all right? So P plus Q equals 1. That's our simple equation. Right now is when we get a little fancier, all right? We've got the frequency of the homozygous dominant trait plus the frequency of the heterozygous genotype plus the frequency of the homozygous recessive equals 1. What does this just mean? Well, if I'm looking at an entire population, guys, we've got to look at all of the homozygous dominant, all of the heterozygous, and all of the homozygous recessive to get a full count of everybody in the group. Does that make sense? You guys already know this. So what the Hardy-Weinberg is, it's taking what you already know and applying it to a group of animals. It's like if we were going to go through a population of chickens and sort them into genotypes for their feather color. All right, we're going to put all of our homozygous dominant chickens in a group, all of our heterozygous chickens in a group, and all of our homozygous recessive in a group. And if we count up those groups all together, it's going to give us the total of the population. Does that make sense? Give me a smiley face on how, like, if that makes sense. I don't like math. I don't like equations, but I like being able to put that into what I do know. I do know that if I'm looking at a group, I know that there are 3 types of genotypes that can be out there, just like 3 colors, right? And I can sort them out. And if I sort them out, they will all fit into one of those 3 categories. And all 3 of those categories together equal our population. Good. It looks like most of us are on board there. Okay? This is what the Hardy-Weinberg is. I'm not going to ask you to solve any equations today. I'm not asking you to solve equations. I'm just asking you to understand what the equation represents. Does that make you feel better too? I'm not asking you to solve it. I'm just asking you to know P is dominant, Q is recessive. And we know, look, P and Q are heterozygous. So our homozygous dominant plus our heterozygous plus our homozygous recessive equal our population. And we can actually plug numbers in for this to solve for allele frequencies. And there are scientists that do that. We are not going to be those scientists today because I want us to just get the understanding of this equation. Now, this only works under basically perfect environmental conditions. All right. It only works if there is random mating. So that means you can't go to a dog breeder and go look at that population of dogs and plug in Hardy-Weinberg. Does that make sense? Because that's a controlled situation. That's not random mating. That means they take 2 dogs of a very specific breed, of a very specific genotype, to get what they want in their population. It's not random. All right. So it needs to be at random. And it needs to be a larger population. We couldn't take like 5 dogs or like 5 chickens and call it a population and plug that in because there's not enough diversity for that to make it accurate. Does that make sense? So we're assuming that they're random mating. We're assuming that there's a big, large population and that equation will work. All right. There cannot be any, it does not calculate for natural selection. It does not calculate for if there's mutations in that population. And there's no calculating as to if one of our Cincinnati chickens came to my Piqua chickens or Alex Kent chickens. All right. You can't have a chicken that's migrated because it could be bringing something new into the equation. So we are assuming that when we use that it's a random mating situation and a large population that's giving us full diversity. Does that make sense? So we can calculate the traits in a population if we know that they're randomly choosing who their mates and we know it's a large population and that none of these weird things are going on, or you have an extra chicken coming in from some crazy place, or a weird mutation, or natural selection. Okay. So we've got our dominant trait, our heterozygous, and our heterozygous recessive and dominant that we're basically sorting out to just say, how frequent is this trait in a population? Well, I can tell you. I can tell you if the population is large enough and there's random mating, we can go in and we can plug in numbers after sorting through our population. Good. Guys, we did it. We got through Hardy-Weinberg today. How do you feel? Could you tell me what an allele frequency in a population is? You could tell me that you can solve that using what equation? How can you find the allele frequency? How can you find how often a trait is? Yes. What equation is that called that we're just talking about today? Hardy. Good. Hardy Weinberg is, we want to tie together with that allele frequency, how often you see a trait in a population. Good job, Kaden. That's impressive. All right, what does P represent? The big P. What does the big P represent? Yep, it represents the dominant trait. What does the little q represent in the Hardy-Weinberg equation? The recessive trait. Good. Tell me two things. There's two factors that the Hardy-Weinberg assumes that has to be true of the population. In order to use that equation, we have to assume that what? Two assumptions that we make. Yes, that there's random mating. Yep. So that they're choosing their spouse randomly. They're not being chosen, like being forced to choose. Good. And a yes, good job, large population, right? We're not just dealing with a very small population with limited choices. Good. Do you feel like we accomplished our goals today? Give me a smiley face if you think we did. And I didn't get us done 10 minutes early, but I got us done 5 minutes early. And we're feeling good, so that makes me feel awesome. Well, I promised you my goal for Valentine's Day for you was to let you out of here early. So that's what I'm going to do. I'm going to get you on your way. Read your lessons, make some flashcards, do some reading, but make sure there are some people in your life that you appreciate today, those people that you love and love you. Happy Valentine's Day, everybody. Thanks for being here. Enjoy your extra few minutes. I know I cheated you 5 minutes. I'll let you out 5 minutes early tomorrow, too. Oh, thank you. Happy Valentine's Day. See ya. Oh, you're wanting to hang around and chat with the extra couple minutes? I'm fine with that. Oh, I'm sorry. Well, the good thing is, I'm gonna try to do some Padlets, Isaiah. Put you in some groups so that you guys can still communicate outside of class and kind of hang out. And my ultimate goal is that you keep each other on track in biology, kind of be accountability, or if you guys want to work together on things, but it can be a place that you go to outside of a neuro classroom where you can still kind of have a social aspect with your classmates.